Every biological function in your body, from fighting off infections to repairing damaged tissue, relies entirely on proteins folding into precise three-dimensional shapes. But mathematically, a standard protein has so many possible configurations that finding its correct shape by random chance would take longer than the age of the universe. Yet in biological reality, proteins fold into their functional states in a matter of milliseconds. This happens because the protein chain is not infinitely flexible. Its movement is strictly governed by the laws of geometry. To see how these laws operate, we have to zoom in past the whole protein, down to the atomic level of its fundamental building blocks, the amino acid sequence. The order we see in biology is not an accident. It is the inevitable result of physical constraints that forbid chaotic, random shapes. At the core of every protein is a chemical spine. This is the polypeptide backbone, a repeating sequence of nitrogen, alpha carbon, and carbon atoms. Look at the peptide bond linking the amino acids. Because it has a partial double bond character, this connection is completely rigid. It cannot rotate, locking these specific atoms into a flat, two-dimensional plane. That rigidity leaves only one swivel point in the chain, the alpha carbon, which is flanked by two single bonds that do allow rotation. But while rotation around that carbon is technically possible, atoms still occupy physical space. Amino acids have bulky side chains, or R groups, creating steric hindrance. If a bond rotates too far, these side chains crash into each other. The three-dimensional architecture of all life is dictated by these microscopic traffic jams, the physical impossibility of atoms overlapping. We have specific names for the two rotating bonds flanking the alpha carbon, the dihedral angles phi and psi. Measuring the exact degree of rotation for both phi and psi gives us a coordinate pair. That pair precisely defines the local shape of the protein backbone. By calculating every possible combination of phi and psi, we can map exactly where atoms will collide and where they can exist peacefully without steric strain. This two-dimensional topographical map of physically possible conformational space is called the Ramachandran plot. Watch what happens if we drag the coordinate dot into a dark, forbidden zone. The three-dimensional model is forced to violently overlap. The Ramachandran plot is a direct mathematical translation of the physical laws governing biology. Notice how the illuminated safe zones on this plot are highly clustered. They are not evenly distributed across the grid. Let's look at a distinct, concentrated cluster down in the bottom left quadrant. When consecutive amino acids fall into the specific phi and psi coordinate range, the backbone coils into a tight, right-handed spiral. This is the alpha helix, the single most common structural element found in proteins. Now shift your focus back to the plot, to the second major illuminated cluster in the top left quadrant. Coordinates in this region cause the backbone to stretch out in an extended, pleated zigzag formation. When multiple strands in this extended conformation align side by side, they bond together to form a strong, flat surface. We call this structure the beta pleated sheet. Biology uses these sheets to create rigid, stable architectures. This diagram of a T-cell surface glycoprotein shows exactly how clear, flat arrays of beta strands are used to form the core of an immune system receptor. The familiar architectural motifs of biology, helices and sheets, are simply the path of least resistance through physical space. But do all 20 amino acids follow the exact same rules on this plot? Consider glycine. Its side chain is just a single hydrogen atom, meaning it has almost no bulk to cause steric hindrance. This lack of physical bulk grants glycine immense flexibility. It can comfortably exist in regions of the plot that are strictly forbidden to every other amino acid. Proline is the exact opposite. Its side chain loops back and covalently binds to the backbone, physically locking its phi angle in place. It can only move up and down along a single axis. The sequence of amino acids in a protein acts as a rigid instruction manual. It dictates exactly which areas of conformational space the chain can access to build its final shape. Moving from the theoretical physics of single amino acids to the reality of modern structural biology means looking at macrostructures containing thousands of atoms. When researchers determine a new protein structure, whether through X-ray crystallography or AI prediction, They have to prove that the resulting model is physically possible. To do that, the Ramachandran plot is used as the ultimate quality control tool. Researchers plot the phi and psi angles of every single amino acid in the entire protein simultaneously. In a perfectly solved, biologically accurate structure, the vast majority of these dots will land safely within the favored regions. The gold standard for validation is strict. A high-quality structure will have over 98% of its residues cleanly within these established safe zones. The plot acts as a forensic audit, a mathematical lie detector for structural models. But we have to look closely at the data points that don't fit, the outliers. These red outliers represent physical impossibilities in the data. They are places where the computational model has mistakenly forced atoms to occupy the same space. Validation software pipelines, like ProCheck, automatically scan the coordinates and flag these geometric violations for scientists to review. Researchers must then iteratively reassign their models, shifting the backbone angles until the strain is resolved and the atoms no longer clash. Whether a protein structure is mapped by a crystal or predicted by an algorithm, it must ultimately obey the absolute